The heart. The heart is a hollow muscular organ that is about the size of a fist. It is surrounded by a thin layer of tissue called the pericardium or heart sac. The main job of the heart is to pump blood through a network of blood vessels. These blood vessels form a loop that starts at the heart, goes out through your body, and then ends up back at the heart again. Together, the heart, blood vessels are part of the circulatory system, which does two important jobs in the body. It carries oxygen and nutrients to all of your cells and picks up carbon dioxide and other waste products that your body produces so they can be disposed of. The inside of a normal heart is divided into 4 chambers: the right atrium, the left atrium, the right ventricle, and the left ventricle. The circulatory loop begins with blood entering the right atrium of the heart. When the heart beats, blood flows from the right atrium into the right ventricle through a valve. In a healthy heart, the valves keep blood flowing in a one-way direction. When they open, they only let the right amount of blood through, and then they close to keep blood from flowing backward in between beats. From the right ventricle, blood flows through another valve and then to the lungs, where it picks up oxygen. From the lungs, it flows back into the left atrium of the heart and through the mitral valve into the left ventricle. From the left ventricle, blood is pumped through the aortic valve and into the aorta, where it goes out to the rest of the body and transfers oxygen and nutrients to your cells. In exchange, the wastes from your cells, including carbon dioxide, are transferred into the blood. As veins carry the deoxygenated blood back to the right atrium of the heart, other organs filter out and dispose of the waste products. After entering the heart, the deoxygenated blood is then pumped through the lungs, where it is loaded back up with oxygen, and the cycle is repeated. If you could see what your heart looks like when it beats, it would look like this. Like all the other tissues in your body, the heart muscles need blood, oxygen, and nutrients to stay healthy. But the heart doesn't get oxygen from the blood that flows through its chambers. Instead, it gets blood and oxygen from the coronary arteries, which wrap around the outside of the heart. The coronary arteries branch several times so that they can reach your entire heart. Since the heart muscles never rest, it needs a constant supply of blood and oxygen. So it's important that blood can easily flow through all the branches of the coronary arteries.
The respiratory system moves air through the nose, pharynx, larynx, trachea, and bronchus to the alveoli, where the gas exchange between oxygen and carbon dioxide occurs. Nares are the openings to the nose. The nasal cavity is lined with cilia, mucous membranes, and blood capillaries. The air is filtered by cilia, moistened by mucous membranes, and warmed by the blood. Air moves into the pharynx or throat, the common passageway for food and air. Air continues on to the larynx. The epiglottis, a flap of tissue in front of the larynx, closes off the larynx when swallowing to prevent food from entering. The larynx or voice box contains the vocal folds. The trachea or windpipe connects the larynx to the bronchial tree. The cartilage rings of the trachea prevent the trachea from collapsing. Lungs are spongy tissue with alveoli and blood capillaries. Breathing occurs because of the expansion and contraction of the lungs. The bronchi carrying the air subdivide into smaller branches called bronchioles. At the end of each bronchiole are the alveolar sacs. The alveolar sacs are surrounded by blood capillaries and contain millions of single-layer alveoli cells where the gas exchange takes place. Oxygenated air goes from the nose to the pharynx, larynx, trachea, bronchus, and alveoli. By the process of diffusion, oxygen in the air moves from the alveoli to the capillaries. Carbon dioxide moves from the capillaries to the alveoli and is exhaled. This process is called respiration.
The foundation for health and recovery from illness rests in the ability of our digestive systems to digest food. All of the nutrients necessary to sustain life reside in the food we eat. Our digestive systems are designed to change food into a form that permits the absorption of those nutrients through the digestive tract, into the bloodstream, and to the organs and systems that depend on those nutrients for life and health. What makes absorption possible are digestive enzymes, most of which are produced in the pancreas and released throughout the digestive tract. Our bodies produce enzymes specific to the type of food we are eating. Protein-digesting enzymes such as protease, the fat-digesting enzyme lipase, and amylase for the digestion of carbohydrates. Food, when eaten, begins to digest in the mouth, but different types of food are digested in different parts of the digestive system. Protein, while masticated in the mouth, is primarily digested in the stomach. Fats are primarily digested in the small intestine. The process of carbohydrate digestion begins in the mouth with the release of the amylase enzyme. In the mouth, amylase enzymes begin to break apart the bonds holding the carbohydrate polysaccharide sugars together. Additional carbohydrate digestion continues from the mouth in the upper stomach. Protein is primarily digested in the stomach. Protein-digesting enzymes break apart the bonds holding the amino acids of protein. Digested protein, partially digested carbohydrates, and undigested fats travel into the small intestine for the digestive process to be completed. In the small intestine, amylase completes the digestion of carbohydrates. Fats are digested in the small intestine by the lipase enzyme. Digested protein, carbohydrates, and fats are then absorbed through the villi of the small intestine into the bloodstream, where crucial life-sustaining nutrients are distributed throughout the body. The foundation for health and recovery from illness lies in the ability of our digestive system to change food into a form permitting absorption of life-sustaining nutrients into the bloodstream. The inability to absorb those nutrients often contributes to a wide range of illnesses. Poor digestion, the malabsorption of nutrients, is endemic today. Constipation, flatulence, heartburn, and acid reflux, so commonplace, are just a few of the indicators of poor digestion. A diet of cooked, processed, and fatty foods, illness, aging, and antibiotics deplete the naturally occurring enzymes in our bodies. Without enzyme supplementation, we risk adversely affecting the biochemical processes so critical to sustaining life. The protease, amylase, and lipase enzymes provided in a capsule of Digest Gold supplement the body's naturally occurring enzymes.
The nervous system is like a network that relays messages back and forth from the brain to different parts of the body. The human brain acts as a central computer that controls all the bodily functions. It is incredibly compact, weighing around just about 3 pounds. Its many folds and grooves provide it with the additional space necessary for storing all the information. The brain is protected by the bones of the skull. It is cushioned by layers of membranes called meninges, as well as a special fluid called cerebrospinal fluid. The brain can be broadly divided into 3 parts: the forebrain, midbrain, and the hindbrain. Forebrain is the largest and the most complex part of the brain. Cerebrum, which is a part of the forebrain, consists of the area with all the folds and the grooves. It contains the information that essentially makes us who we are: our intelligence, memory, personality, emotions, speech, and ability to feel and move. The cerebrum again is divided into 2 hemispheres, left hemisphere and right hemisphere. The left hemisphere is considered to be logical, analytical, and objective, whereas the right hemisphere is thought to be more intuitive, creative, and subjective. The midbrain is located underneath the middle of the forebrain, and it acts as a master coordinator for all the messages going in and out of the brain to the spinal cord. The hindbrain sits underneath the back end of the cerebrum, and it consists of the cerebellum, pons, and medulla. The cerebellum, also called as the little brain, is responsible for balance, movement, and coordination. The pons and the medulla, along with the midbrain, are often called the brain stem. It sends out and coordinates all of the brain's messages. It also controls other automatic bodily functions like breathing, heart rate, blood pressure, swallowing, digestion, and blinking of eyes. There are different centers in the brain which help us perform different activities. The occipital lobe is the center for vision. The movements of the body are coordinated in the cerebrum. The centers located in the temporal lobe are responsible for hearing. The medulla controls the regulation of involuntary activities like heartbeat, breathing, and swallowing. The base of the frontal lobe contains the center which is responsible for speech. The upper part of the precenter sulcus is the center for the movement of leg muscles.
How do nerves work? Are nerves simply the wires in the body that conduct electricity like the wires in the walls of your home or in your computer? This is an analogy often made, but the reality is that nerves have a much more complex job in the body. They are not just the wires, but the cells that are the sensors, detectors of the external and internal world, the transducers that convert information to electrical impulses, the wires that transmit these impulses, the transistors that gate the information and turn up or down the volume, and finally, the activators that take that information and cause it to have an effect on other organs. Consider this. Your mother gently strokes your forearm and you react with pleasure. Or a spider crawls on your forearm and you startle and slap it off. Or you brush your forearm against a hot rack while removing a cake from the oven and you immediately recoil. Light touch produced pleasure, fear, or pain. How can one kind of cell have so many functions? Nerves are in fact bundles of cells called neurons, and each of these neurons is highly specialized to carry nerve impulses, their form of electricity, in response to only one kind of stimulus and in only one direction. The nerve impulse starts with a receptor, a specialized part of each nerve where the electrical impulse begins. One nerve's receptor might be a thermal receptor, designed only to respond to a rapid increase in temperature. Another receptor type is attached to the hairs of the forearm, detecting movement of those hairs, such as when a spider crawls on your skin. Yet another kind of neuron is low-threshold mechanoreceptor, activated by light touch. Each of these neurons then carry their specific information, pain, warning, pleasure, and that information is projected to specific areas of the brain, and that is the electrical impulse. The inside of a nerve is a fluid that is very rich in the ion potassium. It is 20 times higher than in the fluid outside the nerve, while that outside fluid has 10 times more sodium than the inside of the nerve. This imbalance between sodium outside and potassium inside the cell results in the inside of the nerve having a negative electrical charge relative to the outside of the nerve, about equal to minus 70 or minus 80 millivolts. This is called the nerve's resting potential. But in response to that stimulus, the nerve is designed to detect pores in the cell wall near the receptor of the cell open. These pores are specialized protein channels that are designed to let sodium rush into the nerve. The sodium ions rush down their concentration gradient, and when they do, the inside of the nerve becomes more positively charged, about plus 40 millivolts. While this happens initially in the nerve right around the receptor, if the change in the nerve's electrical charge is great enough, if it reaches what is called threshold, the nearby sodium ion channels open, and then the ones nearby those, and so on and so forth, so that the positivity spreads along the nerve's membrane to the nerve's cell body, and then along the nerve's long, thread-like extension, the axon. Meanwhile, potassium ion channels open, potassium rushes out of the nerve, and the membrane voltage returns to normal, actually overshooting it a bit. And during this overshoot, the nerve is resistant to further depolarization. It is refractory, which prevents the nerve electrical impulse from traveling backwards. Then, ion pumps pump the sodium back out of the nerve and the potassium back into the nerve, restoring the nerve to its normal resting state. The end of the nerve, the end of the axon, communicates with the nerve's target. This target will be other nerves in a specialized area of the spinal cord to be processed and then transmitted up to the brain. Or the nerve's target may be another organ, such as a muscle. When the electrical impulse reaches the end of the nerve, small vesicles or packets containing chemical neurotransmitters are released by the nerve and rapidly interact with the nerve's target. This process is called synaptic transmission, because the connection between the nerve and the next object in the chain is called a synapse. And it is here, in the synapse, that the neuron's electrical information can be modulated, amplified, blocked altogether, or translated to another informational process.
Ears are the sense organs for hearing. The human ear mainly consists of three main parts: the external ear, the middle ear, and the internal ear. The external and middle ear are exclusively concerned with hearing. The internal ear has a cochlear part concerned with hearing and a vestibular part meant for maintenance of equilibrium and posture. The external ear consists of an immovable cartilaginous pinna and an external auditory canal leading inwards. The pinna collects and directs sound waves into the external auditory canal. The passageway of the auditory canal is an obstacle course of hairs and wax-producing glands that catch insects, dust, etc. It also helps to moderate the climate inside the ears. The external auditory canal ends at a delicate whitish glistening membranous diaphragm called tympanic membrane. The middle ear is an air-filled chamber on the other side of the tympanic membrane and inside the cranial bone. An eustachian tube emerges from the middle ear and opens into the nasopharynx. It helps in maintaining the balance and air pressure between the two sides of tympanic membrane. The middle ear cavity contains three small pieces of bone, namely malleus or hammer, incus or anvil, and stapes or stirrup. These are called ossicles. Auditory ossicles transmit the sound-induced vibrations of tympanic membrane to the fluid endolymph which fills the internal ear. The internal ear is in the form of a complex system of cavities and is also called labyrinth. It has two organs, namely vestibular apparatus and cochlea. Vestibular apparatus is situated in posterior to the cochlea. It is meant for maintenance of equilibrium and posture and is therefore called a sense organ for balancing. The ear pinna collects and directs the sound waves traveling through air into the external auditory canal. The sound waves create vibrations in tympanic membrane or eardrum. These vibrations are transmitted to the chain of ossicles to the perilymph in the vestibule. With each tiny inward movement of the membrane, the malleus next to it vibrates in the same rhythm and by a lever action transmits the message to the adjoining incus, and the incus in turn transmits the vibrations to the stapes. The stapes bone relays the vibration to the inner ear. The movement of stapes towards the vestibule sets up a pressure wave in the perilymph. The human nose is also known as the organ of olfaction. The fundamental area of smelling in the nasal mucous membrane is called olfactory area. It is brownish yellow in color. The general lining epithelium of the olfactory area is pseudostratified columnar epithelium. Olfactory receptor cells are present within the columnar epithelium which carry the impulse of smell to the brain. There are 10 to 20 million receptor cells in both the nostrils of a man. They are actually a part of the primary olfactory neuron. Receptor cells are surrounded by the several sustentacular or supporting cells. The most peripheral part of the receptor cell is called the olfactory rod. Molecules emanating from the odorant substance are carried by air to the olfactory area and get dissolved in the mucus. In solution, they come in contact with the cilia of receptor cells, and as a result, a receptor potential develops. Finally, through the olfactory nerve, the impulse reaches an appropriate part of the brain and the perception of smell sense occurs. Taste buds are the sense organs for taste sensation. They occur mainly in the mucous membrane over tiny projections called papillae on the surface of the tongue. Each papilla contains several taste buds. Some taste buds are also located in the mucous membrane of the pharynx, palate, and epiglottis. Circumvallate papillae are the largest and circular. They form an inverted V-shaped row at the posterior portion of the tongue. All of them contain taste buds. Fungiform papillae are knob-like elevations over the entire surface of the tongue. Most of them contain taste buds. Filiform papillae are pointed, thread-like structures and are also distributed over the entire surface of the tongue. They rarely contain taste buds. A taste bud is an oval, barrel-shaped structure opening on the tongue surface through a pore called taste pore. It contains 60 to 70 spindle-shaped cells, namely sustentacular cells and taste receptor cells. Taste receptor cells receive taste sensations. Some hair-like processes, known as microvilli, project out of the pore. Substances dissolved in the salivary water flow into the taste bud through the taste pore to reach receptor cells. The receptor cells are thus stimulated, and in turn, the nerve fibers which emerge from the taste buds are also stimulated and eventually reach an appropriate part of the brain to bring about the sense of taste. There are numerous varieties of taste senses, but human beings can generally recognize the four basic tastes: salty, sweet, sour, and bitter. The taste of a substance depends upon structure, function, and interrelationship of molecules. There is no structural difference between taste buds in different areas of the tongue. They differ in their sensitivity to different basic tastes. The tip of the tongue is very sensitive to sweet taste, the back to bitter, and sides to sour and salty tastes. The mid-dorsum of the tongue is insensitive to taste. A baby has many more taste buds than an adult. Some mammals like monkeys and cats also possess taste buds for distilled water. Adaptation of taste sensation is very quick. To conclude, we can say that the major sensory organs of the body, such as eyes, ears, nose and tongue, play a crucial role in our life, which would not be the same in their absence. One of the most important of all sensory organs are the eyes. Eyes are the sense organs for vision and contain receptors called photoreceptors, rod cells and cone cells, which convert the energy or specific wavelengths of light into action potentials of nerve fibers. Photoreceptors give a definite idea about the objects in the environment. Each eyeball is about 2.5 centimeters in diameter. An eyeball is lodged within a bony socket called an orbit. The extrinsic muscle of the eye helps in the movements of the orbit wall. An eyeball consists of three coats. The outer coat consists of the sclera and cornea. The middle coat is called uvea and the inner coat is called retina. The outer wall of the eyeball is formed by a thick, white, opaque membrane called the sclera. It mainly consists of white fibrous tissue known as collagen. Sclera is 5/6 of the outer wall of the eyeball. The region of the outer coat facing directly forward is a transparent cornea. Most of the cornea is made up of a connective tissue called lamina propria. It is 1/6 of the outer wall of the eyeball and has no blood vessels or lymphatics and receives nutrition from vessels around its periphery. Cornea has a rich nerve supply. Light passes through cornea and provides vision. The middle coat is also known as uvea because of its green color and is highly vascular in nature. It has 3 parts. The anterior-most part is known as the iris. The posterior-most part is called the choroid, and the part that lies between the iris and the choroid is said to be the ciliary body. The iris appears as a black screen through the cornea and has a small central aperture called the pupil. It has 2 sets of smooth muscles which are arranged circularly and radically around the pupil. The pupil regulates the amount of light passing into the eye. In bright light, it contracts, and in dim light, it dilates so that the optimum amount of light required for proper vision reaches the retina. The choroid is highly vascular and pigmented and basically helps in the formation of sharp images on the retina. The ciliary body is a ring-like structure continuous with the periphery of the iris. Its posterior part is called the ciliary ring, and it has an anterior part made up of radically arranged ciliary processes. The inner layer of the posterior 2/3 of the eyeball consists of a light-sensitive layer called the retina. It consists of 2 types of photoreceptors, rod and cone cells, and 4 types of neurons, including bipolar nerve cells and ganglion cells. The working of an eye can be compared to that of a camera. Light reflected from an object enters the eye through the pupil. The iris controls the amount of light passing through by controlling the size of the pupil. The rays of light converge as they pass through the aqueous humor, lens, and vitreous humor, and finally focus at the retina, forming an inverted image of the object. The muscle of the eyeball move the image where the vision is sharp, and the lens make adjustments to bring a sharp focus on the retina. The inverted image is then picked up by the optic nerve, taken to the brain, where it is severed, and finally, an upright image of the object is perceived by the individual.
The reproductive system in males and females is responsible for the production of hormones as well as the process of reproduction. One of the most important organs of the male reproductive system are the testicles or the testes. They are enclosed in a sac known as a scrotum. The scrotum helps keep the testicles at a temperature lower than body temperature. This is important since the sperms are produced in the seminiferous tubules located in the testicles. The epididymis is a system of convoluted tubes that connects each testes to the vas deferens. It is responsible for emptying the sperm into the vas deferens. The vas deferens is connected to the seminal vesicle and the prostrate gland. The seminal vesicle and the prostrate gland release secretions that mix with the sperm in order to form semen. The ejaculatory duct carries the sperm into the urethra, located in the penis. The penis contains erectile tissue and becomes erect during sexual arousal. Cowper's gland, located at the base of the penis, prepares the urethra for ejaculation of semen. The female reproductive system is the counterpart of the male reproductive system. The outer genitals consist of the labia majora and the labia minora. The clitoris, like the penis, is composed of erectile tissue. The vagina meets the outer genitalia at the vulva. Just as males have 2 testicles, females have 2 ovaries. The ovaries produce hormones such as estrogen and progesterone. The follicles of the ovaries produce ova or eggs. Every month, 1 mature egg enters the uterine tubes or the fallopian tubes and travels to the uterus. Every month, the endometrium or the lining of the uterus is nourished with blood and fluid in preparation for receiving a fertilized egg. If the egg is not fertilized, it is discharged from the uterus along with the lining. This monthly discharge is known as menstruation.
Menstruation, thus, is the cyclic discharge of blood carrying broken tissue materials through the vagina. The menstrual blood contains stripped of endometrium, mucus, leukocytes, and unfertilized ovum. A menstrual cycle is taken to begin with the onset of menstrual bleeding and ends just before the next menstruation. During the menstrual cycle, major changes occur in the uterus, vagina, breast, and other organs. Also, there is maturation of follicle in the ovary or ovarian cycle. The changes during a menstrual cycle can be divided into 3 phases: proliferative phase, secretory phase, and bleeding phase. After menstruation, the proliferative phase starts with the growth and proliferation of tissues on the walls of the uterus, fallopian tubes, and vagina. Since this phase involves a growing follicle in the ovary, it is also known as follicular phase. The phase extends for 10 to 12 days, by the end of which the ovum is ejected, ovulation from the follicle of the ovary. It involves changes in the uterus and the ovary. At the onset of proliferative phase, the mucous membrane of the uterus, called the endometrium, is the thinnest. The uterine changes then begin due to rising concentration of estrogen. Gradually, endometrium glands grow in length, its epithelial cells proliferate, the endometrial stroma and blood vessels grow. Just before ovulation, the endometrium becomes 3 to 5 millimeters thick. The myometrial contractions become more powerful and secretion of uterine cervix glands becomes very thin during ovulation to facilitate the entry of spermatozoa. During the proliferative phase, an immature follicle ripens into a Graafian follicle. Then comes the secretory phase, extending for the next 12 to 14 days. Here the ruptured follicle changes into a corpus luteum in the ovary and begins to secrete progesterone hormone, which causes changes in the secondary sex organs to further prepare for the anticipated pregnancy. This phase is also known as progesteronal phase or luteal phase. In this phase, the changes in uterus involve further thickening of endometrium. The uterine glands are filled with secretions. Its movements are reduced to keep the uterus non-contracting. The arteries of the endometrium become coiled and its glycogen content increases. Towards the end of this phase, the endometrium is thick, soft, and rich with blood. Next is the bleeding phase, which starts if pregnancy fails to occur. Here the spiral arteries of the endometrium undergo spasm. Hence, portions of endometrium are sloughed off, and this is accompanied by menstrual bleeding. At the end of menstruation, the endometrium that remains is only 0.5 to 1 millimeter thick. The bleeding phase usually extends for 3 to 5 days and is followed by the next menstrual cycle.
The uterus is connected to the vagina by the cervix. During sexual intercourse, the semen from the male penis enters the vagina. The sperm cells then travel to the fallopian tube in order to fertilize the egg. However, it is possible only for one sperm cell to penetrate the ovum or the egg and fertilize it. The genetic material from the sperm and the ovum combine and form the first cell of the zygote or the fertilized egg. The egg then travels to the uterus, where it attaches itself to the endometrium. The fertilized egg develops into a human baby over a period of 40 weeks. During childbirth, the baby is pushed through the cervix and into the vagina through a series of contractions. The vagina serves as the birth canal for the human baby.
There is a whole lot going on inside of a eukaryotic cell. It's more like a city than a monk cell. In fact, let's go with that. A cell is like a city. It has defined geographical limits, a ruling government, power plants, roads, waste treatment plants, a police force, industry, all the things a booming metropolis needs to run smoothly. But this city does not have one of those hippie governments where everybody votes on stuff and talks things out at town hall meetings and crap like that. Nope. Think fascist Italy circa 1938. Think Kim Jong-un's North Korea, and you might be getting a closer idea of how eukaryotic cells do their business. Let's start out with city limits. So as you approach the city of Eukaryopolis, there's a chance that you will notice something that a traditional city never has, which is either cilia or flagella. Some eukaryotic cells have either one or the other of these structures, cilia being a bunch of tiny little arms that wiggle around, and flagella being one long whip-like tail. Some cells have neither. Sperm cells, for instance, have flagella and our lungs, and throat cells have cilia that push mucus up and out of our lungs. Cilia and flagella are made out of long protein fibers called microtubules, and they both have the same basic structure. 9 pairs of microtubules forming a ring around 2 central microtubules. This is often called the 9+2 structure. Anyway, that's just so you know when you're approaching the city, watch out for the cilia and flagella. If you make it past the cilia, you will encounter what is called a cell membrane, which is a kind of squishy, not rigid plant cell wall, which totally encloses the city and all of its contents. It's also in charge of monitoring what comes in and out of the cell, kind of like the fascist border police. Each eukaryotic cell is filled with a solution of water and nutrients called cytoplasm. And inside of this cytoplasm is a scaffolding called the cytoskeleton. It's basically just a bunch of protein strands that reinforce the cell. Centrosomes are a special part of this reinforcement. They assemble long microtubules out of proteins that act like steel girders that hold all the city's buildings together. The cytoplasm provides the infrastructure necessary for all the organelles to do all of their awesome, amazing business, with the notable exception of the nucleus, which has its own kind of cytoplasm called the nucleoplasm, which is a more luxurious, premium environment befitting the cell's beloved leader. But we'll get to that in a minute. First, let's talk about the cell's highway system. The endoplasmic reticulum, or just ER, are organelles that create a network of membranes that carry stuff around the cell. These membranes are phospholipid bilayers, same as in the cell membrane. There are two types of ER. There's the rough and the smooth. Fairly similar, but slightly different shapes, slightly different functions. The rough ER looks all bumpy because it has ribosomes attached to it, and the smooth ER doesn't, so it's a smooth network of tubes. Smooth ER acts as a kind of factory warehouse in the cell city. It contains enzymes that help with the creation of important lipids. Other enzymes in the smooth ER specialize in detoxifying substance, like noxious stuff derived from drugs and alcohol, which they do by adding a carboxyl group to them, making them soluble in water. Finally, the smooth ER also stores ions in solutions that the cell may need later on, especially sodium ions, which are used for energy in muscle cells. So the smooth ER helps make lipids, while the rough ER helps in the synthesis and packaging of proteins. And those proteins are created by another type of organelle, the ribosome. Ribosomes can float freely throughout the cytoplasm or be attached to the nuclear envelope, which is where they're spat out from. And their job is to assemble amino acids into polypeptides. As the ribosome builds an amino acid chain, the chain is pushed into the ER. When the protein chain is complete, the ER pinches it off and sends it to the Golgi apparatus. And the city that is the cell, the Golgi, is the post office, processing proteins and packaging them up before sending them wherever they need to go. Calling it an apparatus makes it sound like a bit of complicated machinery, which it kind of is, because it's made up of like these stacks of membranous layers that are sometimes called Golgi bodies. The Golgi bodies can cut up large proteins into smaller hormones and can combine proteins with carbohydrates to make various molecules, like for instance, snot. The bodies package these little goodies into sacs called vesicles, which have phospholipid walls just like the main cell membrane, then ship them out either to other parts of the cell or outside the cell wall. We learn more about how vesicles do this in the next episode of Crash Course.
The Golgi bodies also put the finishing touches on the lysosomes. Lysosomes are basically the waste treatment plants and recycling centers of the city. These organelles are basically sacs full of enzymes that break down cellular waste and debris from outside of the cell and turn it into simple compounds which are transferred into the cytoplasm as new cell-building materials.
Now, finally, let us talk about the nucleus, the beloved leader. The nucleus is a highly specialized organelle that lives in its own double-membraned, high-security compound with its buddy, the nucleolus. And within the cell, the nucleus is in charge in a major way. Because it stores the cell's DNA, it has all the information the cell needs to do its job. So the nucleus makes all the laws for the city and orders all the other organelles around, telling them how and when to grow, what to metabolize, what proteins to synthesize, how and when to divide. The nucleus does all this by using the information blueprinted in its DNA to build proteins that will facilitate a specific job getting done. For instance, on January 1st, 2012, let's say a liver cell needs to help break down an entire bottle of champagne. The nucleus in that liver cell would start telling the cell to make alcohol dehydrogenase, which is the enzyme that makes alcohol not alcohol anymore. This protein synthesis business is complicated. Lucky for you, we will have, or may already have, an entire video about how it happens. The nucleus holds its precious DNA along with some proteins in a web-like substance called a chromatin. When it comes time for the cell to split, the chromatin gathers into rod-shaped chromosomes, each of which holds DNA molecules. Different species of animals have different numbers of chromosomes. We humans have 46, fruit flies have 8. Hedgehogs, which are adorable, but less complex than humans, have 90. Now the nucleolus, which lives inside of the nucleus, is the only organelle not enveloped by its own membrane. It's just a gooey splotch of stuff within the nucleus. Its main job is creating ribosomal RNA, or rRNA, which it then combines with some proteins to form the basic units of ribosomes. Once these units are done, the nucleolus spits them out of the nuclear envelope where they are fully assembled into ribosomes. The nucleus then sends orders in the form of messenger RNA, or mRNA, to those ribosomes, which are the henchmen that carry out the orders in the rest of the cell. How exactly the ribosomes do this is immensely complex and awesome. So awesome, in fact, that we're going to give it the full crash course treatment, an entire episode. And now for what is, totally objectively speaking, of course, the coolest part of the animal cell. Its power plants, the mitochondria, the smooth, oblong organelles where the amazing and super important process of respiration takes place. This is where energy is derived from carbohydrates, fats, and other fuels and is converted into adenosine triphosphate, or ATP, which is like the main currency that drives life in Eukaryopolis. You can learn more about ATP and respiration in an episode that we did on that. Now, of course, some cells, like muscle cells or neuron cells, need a lot more power than the average cell in the body, and so those cells have a lot more mitochondria per cell. But maybe the coolest thing about mitochondria is that long ago, animal cells didn't have them, but they existed as their own sort of bacterial cell. And one day, one of these things ended up inside of an animal cell, probably because the animal cell was trying to eat it. But instead of eating it, it realized that this thing was really super smart and good at turning food into energy, and it just kept it. It stayed around. And to this day, they sort of act like their own separate organisms, like they do their own thing within the cell, they replicate themselves, they even contain a small amount of DNA. Now, what may be even more awesome, if that's possible, is that mitochondria are in the egg cell when an egg gets fertilized. And those mitochondria have DNA. But because mitochondria replicate themselves in a separate fashion, it doesn't get mixed with the DNA of the father. It's just the mother's mitochondrial DNA. That means that your and my mitochondrial DNA is exactly the same as the mitochondrial DNA of our mothers. And because this special DNA is isolated in this way, scientists can actually track back and back and back and back to a single mitochondrial Eve who lived about 200,000 years ago in Africa.
Every human being starts out the same way. 2 cells, one from each parent, found each other and became one. And that one cell reproduced itself, dividing and dividing and dividing, until there were 10 trillion of them. Do you realize there's more cells in one person's body than there are stars in the Milky Way? But those 10 trillion cells aren't just sitting there in a big pile. That would make for a pretty boring human being. So what is it that says a nose is a nose and toes is toes? What is it that says this is bone and this is brain and this is heart and this is that little thing in the back of your throat you can never remember the name of? Everything that you are or ever will be made of starts as a tiny book of instructions found in each and every cell. Every time your body wants to make something, it goes back to the instruction book, looks it up, and puts it together. So how does one cell hold all that information? Let's get small. I mean really small, smaller than the tip of a sewing needle. Then we can take a journey inside a single cell to find out what makes up the book of you, your genome. The first thing we see is that the whole genome, all your DNA, is contained inside its own tiny compartment called the nucleus. If we stretched out all the DNA in this one cell into a single thread, it would be over 3 feet long. We have to make it fit in a tiny compartment that's 1,000,000 times smaller. We could just bunch it up like Christmas lights, but that could get messy. We need some organization. First, the long thread of DNA wraps around proteins clustered into little beads called nucleosomes, which end up looking like a long beaded necklace. Then that necklace is wrapped up in its own spiral like an old telephone cord, and those spirals get layered on top of one another until we have a neat little shape that fits inside the nucleus. Voila! 3 feet of DNA squeezed into a tiny compartment. If only we could hire DNA to pack our suitcases. Each tiny mass of DNA is called a chromosome. The book of you would have 46 chapters, 1 for each chromosome. 23 chapters of your book came from your mom, and 23 chapters came from your dad. 2 of those chapters, called X and Y, determine if you're male, XY, or female, XX. Put them together and we get 2 almost identical, but slightly different sets of 23 chapters. The tiny variations are what makes each person different. It's estimated that all the chapters together hold about 20,000 individual instructions called genes. Written out, all those 20,000 instructions are 30 million letters long. If someone were writing 1 letter per second, it would take them almost an entire year to write it once. But it turns out that our genome book is much, much longer than just those 30 million letters. Almost 100 times longer. What are all those extra pages for? Well, each set of instructions has a few pages of nonsense inserted that have to be taken out before we end up with something useful. The parts we throw out we call introns. The instructions we keep we call exons. We can also have hundreds of pages in between each gene. Some of these excess pages were inserted by nasty little infections in our ancestors, but some of them are actually helpful. They protect the ends of each chapter from being damaged, or some help our cells find a particular thing they're looking for, or give the cell a signal to stop making something. All in all, for every page of instructions, there's almost 100 pages of filler. In the end, each of our book's 46 chapters is between 48 and 250 million letters long. That's 3.2 billion letters total. To type all that copy, you'd be at it for over 100 years. and the book would be over 600,000 pages long. Every type of cell carries the same book, but each has a set of bookmarks that tell it exactly which pages it needs to look up. So a bone cell reads only the set of instructions it needs to become bone. Your brain cells? They read the set that tell them how to become brain. If some cells suddenly decide to start reading other instructions, they can actually change from one type to another. So every little cell in your body is holding on to an amazing book full of the instructions for life. Your nose reads nose pages, your toes read toes pages. And that little thing in the back of your throat? It's got its own pages too. They're under uvula.
Your body is made up of 50 trillion cells. Cells come in many different varieties, with many different functions. But inside almost every cell is a nucleus, containing 99.9% of your genes, and mitochondria, containing a few more genes. All told, you have nearly 20,000 genes. Your genes are small parts of a long molecule called DNA, or deoxyribonucleic acid. If you lined up all of the DNA containing all of your genes, it would measure 6 feet long. But it's coiled so tightly that it fits in just one cell nucleus. DNA is a double-stranded molecule composed of sugar, phosphate, and 4 different bases: adenine, thymine, cytosine, and guanine. These bases spell out the language known as the genetic code. The number and order of these 4 bases determine, for example, whether you are a chimp, a cow, a banana, or a human. Most genes are recipes for making specific proteins. These recipes are passed down from parents to children, from generation to generation. When someone says, "You have your father's hair," what they mean is, you appear to have inherited a gene or genes from your father that makes a protein that instructs your hair follicle cells to produce hair that curls like your father's. But they usually opt for the shorter version. Genes tell a cell how to function and what traits to express. More specifically, gene regulators turn different genes on and off in different cells to control cell function. The long molecules of DNA containing your genes are organized into pieces called chromosomes. Different species have different numbers of chromosomes. Humans usually have 46 chromosomes, 2 sets of 23, or simply 23 pairs of chromosomes. Chimpanzees have 2 sets of 24, or 24 pairs of chromosomes. Rhesus monkeys have 21 pairs of chromosomes. Cows have 30 pairs of chromosomes. Chickens have 39 pairs. Fruit flies have 4 pairs, and bananas have 11 pairs. So, what percentage of the DNA in your chromosomes do you share with other species? You share 93% of your DNA with the rhesus monkey and 98.5% with our friend chimpanzee. How about with other humans? 99.5%. So, what makes us different from one another? An entire set of 23 human chromosomes is called a genome. The human genome is composed of 3 billion base pairs. Variation at a single base pair is called a SNP, or single nucleotide polymorphism. When the body makes new cells, it doesn't make many mistakes. But nobody's perfect. Sometimes, when the genome is copied to make a new cell, a single base pair gets left out, added, or substituted. Single base pair substitutions create SNPs. There are around 10 million SNPs in the human genome, which account for many of the genetic differences between you and everyone else on the planet. Some SNPs account for differences in appearance. Others can affect how we develop diseases or respond to drugs. Most SNPs, however, seem to lead to no observable differences between people at all. Since variants are passed down from one generation to the next, the number of differences between your DNA and your neighbors can tell you how closely you are related to each other. Your genes come from your parents, of course, and from their parents, and from their parents' parents, and, well, you get the idea. You have 2 sets of 23 chromosomes, 1 set from each parent. For almost every chromosome you inherit from your mother, you also inherit one from your father that has his versions of the same genes. Together, they form a pair of homologous chromosomes. The X and Y chromosomes, however, are special. Usually, females have 2 X chromosomes, while males have an X and a Y. You get one of your X chromosomes from your mother. Whether you get your father's X chromosome or his Y chromosome determines your sex. Most adult cells contain 2 sets of chromosomes, but sperm and egg cells have only 1 set of 23 chromosomes each. When the body forms sperm or egg cells, a cell divides and pairs of chromosomes separate. A random member of each pair moves into each new cell. This is why, when you were conceived, you obtained half of your mother's genes and half of your father's genes. But your siblings didn't necessarily get the same versions of your parents' genes that you did. Unless you happen to be an identical twin. To form sperm or egg cells, your chromosomes double, like so. When the homologous pairs separate, sometimes they cross over, and at seemingly random points, exchange DNA. This is called genetic recombination. Because your genes get shuffled during recombination, the chromosomes you pass along to your children are not exactly the same as the ones you inherited from your parents. This makes it hard to use most of your chromosomes to trace your genealogy back very far. However, most of the Y chromosome is handed down from father to son entirely intact, Likewise, in humans, DNA in the mitochondria is passed down only from mother to child. For this reason, ancestry along your father's line or your mother's line is easier to trace using the Y chromosome or mitochondrial DNA. When sperm and egg cells join together at fertilization, they create a single cell with two complete sets of 23 chromosomes, one set from your mother and one from your father. This single cell will divide to create new cells over and over until it creates a child. Your observable traits, also known as your phenotypes, result from the interaction between your genes and the environment. This interaction begins in the womb and continues throughout your life. Differences in some phenotypes, like height, are determined mostly by genes. If you have short parents and grandparents, you probably don't tower over your peers, though environmental factors like nutrition can have some effect on your height. Genes also play a role in whether you have a healthy body weight. But diet and exercise can profoundly affect how much you weigh. How genes influence your personality is less understood. You can now learn about over 1 million SNPs in your genome all at once. And every day, scientists are learning more and more about how some of these SNPs affect your phenotype. Getting to know your genome can help you understand a little better why you are the way you are, and in what ways you're similar to or different from your family, friends, and neighbors.